The first rule of biotech is control some IP somewhere. And in radiopharmaceuticals, if you can't control the supply chain, IP is the next best thing. I'm Rachel Williamson, and this is Phase 3. The radiopharmaceutical sector is increasingly looking like a two-hander. You make or license the biological tracker and you lock down the isotope supply, hopefully locking out competitors in the process. But what if you don't need to lock down the isotopes because you have the only IP that can make them work? Or what if instead you're building an intellectual property bank, one that looks like it might prove very attractive to a buyer before you have to make the call on isotope supply? That's the question we'll be dancing around in this episode. And we're going to start with Clarity Pharmaceuticals, a company that prides itself for being born and bred in Australia. Executive Chair Dr. Alan Taylor says their copper isotopes are similar to lutetium, the star of the two radiopharmaceutical therapies on the market today, Lutathera and Pluvicto, both owned by Novartis. But the copper isotopes are easier to make because instead of needing a whole nuclear reactor, they use a rototron, which is like a cyclotron but lower powered. And Alan says no one else has their special tech that can lock copper isotopes onto the biological tracker. So no one else is really using them. And that means their economic moat is in that technology, not in their access arrangements for isotopes. It also means they don't really have to compete for the isotopes. Yet. Alan, tell us where your science came from.
When we talk about biotechs, we talk about their intellectual property, usually. We talk about their smarts, their know-how, and where they've actually come up with that capability. So when I say 100% homegrown from the benchtop, that means the cage that I'm referring to was developed in Australia some 70 years ago in the first instance. Then that fell into the hands of Professor Paul Donnelly at the University of Melbourne. Paul did a few things with it, made it easy to use and easy to conjugate, those sorts of things, but importantly made it bifunctional, that it not only held the metal, but we could actually attach anything to it, from a large antibody to a small molecule. And then, only about 5 or 6 years ago, now I've been involved in Clarity for some 11 years, we went about developing a PSMA product. The BIS PSMA is our major asset within our company.
As a reminder for our listeners, PSMA is the abbreviation of prostate-specific membrane antigen, a protein biomarker found on prostate cancer cells.
It is. But at first we made one like the others. We made one like Pluvicto and Polarify and Gallium PSMA, the generic products. And ours was equally as bad as theirs.
Why do you say bad?
Because it had modest uptake and poor retention in lesions, and that's not good for therapy nor diagnostics. So we made a new one at the benchtop, and we made this bispecific that increased the uptake 2 to 3 times into the lesions. It was retained over the time. A lot more was internalized. It was in collaboration between Clarity and the University of Melbourne that we did that. We did that through some industry academic grants, and we funded people within the lab of Paul Donnelly, and continue that relationship on today and wrap around that an incredible team, the majority of which are here in Australia. And now we have about a third of our team in the US help run our clinical trials and our ops and our supply chain.
You've got 7 clinical trials underway right now and 24 milestones on the calendar between now and 2026.
That would be around about right. I'm sure we could do more.
The trials are for diagnostics and treatments for prostate cancer, neuroblastoma, which affects children and starts out in the nerves, and the rare neuroendocrine cancer, which is a cancer of the nerves and glands. Clarity's thing is that the 12.7-hour half-life of copper-64 means you can run a scan on a person twice and pick up more tumour cells. Alan, you're saying that improves the sensitivity of the test, so tell me about these clinical trials and how that thesis is playing out.
I want to come back and reflect on our tech. The product we make is a diagnostic and a therapy. It relates to the isotope we use. and this is where we're very unique in the marketplace. When we use copper 64 with our product, it's a diagnostic. And then we can utilize copper 67 with our product, and it's a therapy. So it is chemically the same product for diagnosis and therapy. And this is because we're utilizing this perfect pairing of copper isotopes. So I'm going to come back to SAR-BIS PSMA because that's the one that's really taken off. SAR-BIS PSMA is in phase 3 clinical trials as we speak.
I'll pop in here and explain some of those acronyms. SAR stands for sarcophagene, and that is the cage Alan referred to earlier that holds the copper isotopes. The cage is the chelator, the link bonding the isotopes and the biological tracker together. And BIS means twice or double, as Clarity uses 2 biological targeting agents. It says preclinical data shows that this delivers more of the isotope into tumours. Back to you, Alan.
Yeah. And so that product, diagnostically, so the use of copper 64 has already kicked off phase 3 clinical trial in pre-prostatectomy. So before a man, he goes in, he's got rising PSA, they detect cancer in his prostate, and we use PSMA then to image before the prostatectomy and to see whether there's, firstly, look at the lesion and then look if there's any metastatic disease. Now, the problem with the market, Telix's product, gallium PSMA 11, and also the Pylarify product, is that these products have very high, what's termed specificity, but have very low sensitivity. Now, sensitivity is incredibly important, particularly in this disease, because specificity means if it lights up, it's got a high probability of being cancer. But when something's got low sensitivity, it means that if it doesn't light up, it doesn't mean it's not cancer. And these agents are as low as 25% up to about 50%, particularly in the latest one, Posluma. And so you're missing a lot of cancers.
And I guess that kind of data isn't really good enough for a man who's about to have a prostatectomy. For our listeners, that's surgery to have your prostate out, and it comes with the risks of impotence and incontinence. So that's the phase 3 trial for before a prostatectomy. Alan, I understand you're also recruiting for a phase 3 with the same product and the same cancer, but for men who have already had that surgery or treatment?
We're looking to kick off the second phase 3 with the same product so we can treat the lesions earlier and actually have better outcomes. Now, in the phase 2, we found that we could find much smaller lesions than the current standard of care, the gallium PSMA product and the Pylarify products. And we went head-to-head on some of those.
How small?
We're detecting lesions down as small as diameters smaller than 2 millimeters. So that phase 3 will begin recruiting this year. But that same product, again, is a therapy. And we're doing the secure trial, and we're in a dose escalation, dose expansion trial of that. We kicked off at very low doses, and we started to see, even at low doses, a therapeutic benefit for a third of the patients. We went to the next stage, which is about the equivalent dose of Pluvicto at what's termed 8 gigabecquerels, and we found all 3 patients had a phenomenal PSA response. And one of those patients, under what's called expanded access, had what's termed a complete response after a second dose. This is a patient who'd failed 5 to 7 lines of therapy and had no other hope. And suddenly we had that response in the first patient we ever treated twice with what we thought was a therapeutic dose. We've moved that to 12 gigabecquerels because we're doing a dose escalation safety trial. This was only meant to be safety, but there's a lot of efficacy that we're presenting. And at 12 gigabecquerels, we found, once again, great responses in PSA.
We've talked about the isotopes you're using, but what about the biomarkers you're using?
Yeah, so it's quite funny. And I know we started with isotopes. I would call that back to front. The real science is the molecule. PSMA is the first one I mentioned, and we created a new product, the bispecific with 2 targeting moieties. The next one in line is a product called Bombesin. And Bombesin actually derives from a frog, of all things, and was derived by a scientist. And I'm going to, once again, wax lyrical about the science. A scientist called Helmut Macker. He had made a couple of molecules utilizing Bombesin and then collaborated with Paul Donnelly again at very early stages and produced what he termed as the best bombesin he'd ever created. And because of the charge between our unique chelator, the cage, and the charge of bombesin. And it actually targets the gastrin-releasing peptide receptor.
And what is that?
That is a receptor that's expressed on a number of different tumours. So that means it becomes a pan-cancer target. So we started off in breast cancer. We generated some great data there. But we had the urologist calling us up, asking to utilize the product in prostate cancer in patients that were PSMA negative. And that went really well. And then the third product, which is, you touched on neuroblastoma. So we're moving that at the moment in therapy trial with a diagnostic alongside it, only in the US, because in childhood cancer, the great thing is there aren't lots of kids dying of cancer. The bad thing is it then becomes very difficult to run clinical trials because of the small numbers.
Your copper isotopes are beta emitters. Beta emitters generally have a couple of decades of safety data on them. Maybe not copper, but generally. Does that make a difference when you're treating children?
We think the betas have a great opportunity for going much earlier stage, much younger patients that you can treat. We're treating kids and up to very, very high doses of radiation, not having significant side effects, which is wonderful. But it is barely scratching the surface of a massive oncology market. And then we're looking to move this obviously into combination therapies like IO, like Keytruda is a blockbuster drug, and also best standard of care. So in prostate cancer, getting the first line therapies with these new androgen receptor inhibitors and the like. It is just at what we think is the start of the frontier in this space. And we're excited to be there.
That was Clarity Pharmaceuticals Executive Chair, Dr. Alan Taylor. Clarity prides itself on being a homegrown, from the lab upwards Aussie company, and it is using that unique IP as a moat not only around its science, but around its isotope supply chain as well. Our next guest's company is doing things a little bit differently.
Hi there, I'm Charis Palmer, producer of Phase 3. When Rachel and I set about building a new podcast for life science leaders, scientists and long-suffering biotech investors, we looked at what was missing in this space. We believe Phase 3 serves an unmet need for in-depth conversations in a world where nuance matters and AI-written investment articles simply won't cut it. If you agree, please follow us and sign up to our newsletter via LinkedIn. Pledge financial support at phase3.buzzsprout.com and rate and review the podcast on the podcast platform you use to help bring it to the attention of others. Now back to the show.
If Clarity is building out Aussie ideas, Radiopharm Theranostics is licensing in. It's bought in IP working with 5 of the most common isotopes, from gallium-68 to lutetium, and even some of the more speculative ones like terbium-161. The trackers are a gamut of small molecules, peptides, monoclonal antibodies, and nanobodies. And the cancers it wants to find or destroy include prostate, of course, breast, brain, and pancreatic. It's quite the portfolio, and one that has attracted the eye of a very big US pharma company during its recent capital raise. But it's also sold a big dream very early in its life, and investors are displeased. So I asked the man who brought the IP together, Executive Chairman Paul Hopper, about how he's positioning Radiopharm Theranostics during the global race for supply chain security. I've got to address the elephant in the room. You've got some fairly annoyed shareholders from the looks of it who have watched the share price drop from 60 cents when it IPO'd in 2021, and then the capital raising this year at 5 cents. I know this was a premium to the 3 cents where it's been, but what is your message to them?
Yeah, look, so we are acutely aware of that, and that's weighed very heavily on the board and management. I mean, and the board and management are all invested. I mean, up until yesterday at the shareholders meeting, I was the biggest investor, so I certainly feel their pain. The IPO timing, there was no way we could predict that, interest rates would go up and the market would fall out of bed. So we listed in November 21, and on the day one of the IPO, the stock performed badly. And then, of course, we did raises at smaller share prices and so on and so forth. And I think there was a view in the market that the capital demands of the business were high, and that acted as a very bad drag on the share price. And then last year, to be frank, we missed a couple of targets that we said we were going to do, and that all combined to just drive the share price down. So to those people that are still in it, and all of the board and the management and myself, of course, we're still there, is I think we've turned the corner. We've now got funding well into 2026, which will fund all the programs. And what we did last year was we had to put a lot of the programs on go slow. We didn't stop them, but we certainly slowed things down. And I got the CEO to make really serious reductions in the headcount. That's all behind us now. We've got the premier radiopharmaceutical company in the world going to take maybe up to 19.9%. We have a very collaborative relationship. The two assets that we transferred to them as part of the deal, they actually would like us to help them develop them. So I think the future looks good. I mean, obviously, I haven't looked at it today, but whatever the price is, 4 cents, it's a long way back to 60 cents, but the company is in very good shape today.
Let's talk about this in the context of Lantheus, which you now have on your register, your share register, as a 7% shareholder. For our listeners, Lantheus is a major US radiodiagnostics and therapy company and they're worth $6.6 billion-ish US dollars. Paul, I'm really keen to get your take on how to understand this investment. Is it IP related? Is it the fact that you've got 3 clinical trials underway and there's licensing opportunities if they perform as you expect? Or is it because it kind of looks like big US companies are chasing down anything that moves right now and you've got some traction with your science?
Yes, so we at Radiopharm have known them for 3 or so years since our IPO because we shared a technology. In those days, they were purely an imaging radiopharmaceutical company, not a therapeutic. And so they owned the imaging side of one of our technologies and we owned the therapeutic. So we knew them quite well. And when we were raising money recently, we invited them into the raise and they were very keen to get a foothold. So look, I think it was an indication that they liked the whole of the portfolio. Otherwise, they could have quite easily come to us and said, oh, we like your pancreatic radiopharmaceutical, or we like your brain metastasis. And that would often be the normal case that a big pharma, and I sort of call them a big pharma because at 7 or 8 billion, they're getting into that range. They could have just plucked one of the assets out and done a licensing deal, but they decided to invest in the company, which basically, I guess, gave them exposure to every one of the assets we have, of which there are 6. Phase 1, phase 2, or about to go into phase 1 and 2 preclinical.
They did get 2 of your assets. So I understand they paid, was it $3 million for 2 assets? Was that correct?
Yes. So as part of the transaction, it's a little bit hard to sort of ascribe the $3 million, which, as you correctly point out, they paid. They looked at it more of a package that they would invest $10 million US. And as part of that, they would like to get 2 of these preclinical assets which we weren't developing. And we said, well, we have actually spent some money on those 2 assets, not a lot. And they said, well, there's some historical costs. We'll give you that back. So they gave us $3 million.
I see. Something we're going to cover in a later episode in this series is intellectual property and how radiopharmaceuticals companies are creating serious IP portfolios. And part of the reason they're doing this is because the acquisitions market for companies in this space have been really kicking off. I'm interested to know what you think of this in relation to Radiopharm.
Yeah, sure. So we did not develop any of the technologies in our portfolio ourselves. We acquired them or in-licensed them from either universities or medical institutions that had developed them. So with that approach, you don't own the IP, you get a license to the IP. So if the IP is doubtful or not clear or not robust or has 12 months left on it, we don't license it. So the IP, you sort that almost out from the day one that you find the technology that you like. If the IP doesn't stand up to diligence or close scrutiny, then that's the end of the discussion. Because as you know, in biotech companies, we generally don't own bricks or mortar. The asset we own is the right to the technology, which is protected through the intellectual property.
Let's talk about the NASDAQ listing. So
Sure.
Radiopharm promised to start a listing last year, then pulled it because the market conditions weren't good. Now it's on for the end of this year. What's the reasoning behind listing in the US, especially at such an early stage of the company's life when you've got, so far to go with those clinical trials?
Yes, so it's a very simple answer. As part of the 70 million raise we did last month, there were 5 US institutional investors that came in on the fundraising, and one of their requirements was that we do a level 2 compliance listing. So this is not a full.
Why do they want you to do that when they can clearly invest also in you when you're Australia listed?
Yes, I think they wanted to have a US ticker symbol in the sense that if they did want to trade on the US, they can. It wasn't. I can't be any more prescriptive than that. That was one of the conditions that the bankers brought to us, and provided it wasn't a level 3 full IPO listing with the capital raise, with all the Sarbanes-Oxley and the governance required, we were happy to do it. So, and as you pointed out, we'd actually flagged this 18 months ago, so a lot of the work had actually been done. So we will get that listing before Christmas. It's pretty much completed now.
So much of what pharmaceutical companies actually need is in the US. All these companies I'm talking to have bases in the US where they are securing their isotopes. And on the pharmaceutical side, obviously, you're having to go through the Federal Drug Administration in the US or the FDA for your key approvals. And I understand even your CEO is based in the US. It seems like the US might have been a better fit. So why did you list? I guess the better question might be, why did you list in Australia?
Sure. So I've listed all my companies, all my biotech companies on the ASX because it can be a little easier than going out for a straight IPO on NASDAQ or whatever exchange in the US. You can list a slightly smaller company at times, and often the appetite for early stage biotech is a little higher in Australia. I wouldn't say so much nowadays, but at the time you could launch IPOs, up until 2019, 2021, in the era of free money, the market was pretty buoyant. But when that all changed, interest rates went up and technology was relegated to the basement with biotech under that, it became much harder. So the ASX has been a good venue to list companies.
It feels like the tide is turning on that just this year. Do you think those buoyant times might be coming back for biotechs?
Yeah, so there's definitely, well, as the Americans say, there's definitely green shoots in the biotech market that you can see happening. There's been some big raises. I mean, Clarity raised $100+ million 3 or so months ago. We raised 70 off a market cap of 15. So that's like 4 times your market cap, which is pretty much unheard of. So yes, I think there is a stirring there, which is great.
Aside from the NASDAQ listing, what are the big milestones on the horizon from an investment and business perspective that we should be looking out for?
Right. So we are a company that has 4 therapeutic drugs and 2 imaging. I think the consensus is that the market looks at therapeutic radiopharmaceuticals, that is the treating of the cancer, as more attractive than just taking images of it. So we've got a pancreatic cancer drug. Our 4 priority candidates have had approximately 172 patients treated in them across the 4 technologies. So we're not really worried so much about what you normally look for in phase 1, which is safety. Because, for example, our pancreatic cancer has had 99 patients treated in Europe under compassionate use. So we don't think there's a safety issue. So we've got a number of announcements or data releases that are going to come out over the next 6 to 12 months. In the pancreatic study, we hope to dose the phase 1 in America, in New York. It's only 9 patients, it's only small. We hope to get that done in the next number of months. We have a technology from London, from Imperial College, that I license for metastatic brain cancer. So that is primary cancers like breast or whatever that have metastasized to the brain. That's been in 41 patients already in London. And we're looking to announce some data on the phase 2B, again in the next 6 months or so. And then we have these two little antibodies that are called nanobodies here in Australia for PDL1. The target is PDL1 in lung cancer. And we hope to have a readout on some of that on some patients before Christmas. And then the first cousin to that is another nanobody for HER2, which is a very common target in breast cancer and gastric cancer. And we're just negotiating with a few centres in Australia to start a study before Christmas. So there's a good number of announcements in the next 6 months.
That was Radiopharm Theranostics Executive Chairman Paul Hopper. Science IP could be the other way to play the radiopharmaceuticals game, instead of also locking down isotope supplies, that is. It will take time to see how this plays out. In our next episode, we'll take a look at whether building an Australian supply chain is possible. From mine to medicine, as one report dubbed it. Australia has the tech, or almost has the tech, to handle all parts of the chain, from recycling nuclear waste into cancer treatments to delivery. But given the head start that the US and Europe already have, and given the global authority the health regulators in those areas have for approving drugs, we shall see.